So now I want to talk a little bit about DNA replication. And this, I don't know, this is just sort of a funny slide, right? I think you're all familiar with the power of and I just turned the and around because it certainly is one of my favorites, is to talk about DNA and its structure and how it works. And of course, what we've been talking about up to this point, genetics is certainly part of that. So I want to start talking first about the chemical structure of DNA, a little bit about the discovery of it, and we've already talked about the structure of DNA when we were talking about nucleic acid structure way at the beginning of the semester. So just a little reminder on that, but let me start by talking about the discovery of the structure of DNA. So these three guys here, we have James Watson, which maybe some of you have heard of him before. He's the only one of the three that's still living. And Francis Crick and Maurice Wilkins. I think many people hear about Watson and Crick, but we forget about poor old Maurice. And they were the three who were important for discovering the structure of DNA. And they actually did this back in 1953. And these three received the Nobel Prize for uncovering and discovering basically how DNA was built back in 1962. And Watson and Crick, again, are generally thought of as the primary contributors to this. And they did a lot of their thinking about this by model building. And so in the next slide, there's kind of an interesting picture of the two of them looking at their model. So here are James Watson and Francis Crick, and I have arrows pointing to who's who. And just to put this in a little perspective for you, I think that James Watson, at the time that he was doing this work and working with Crick on this, I think he was like 25 years old. So he was a very young man, and Crick was only 35 years old. So can you imagine doing this at age 25, something that turns out to be this important? It's quite something. So you might not be able to see it in this photo very well, but they were working with, the materials that they had. So they were making, metal models of the possible structure of DNA and trying to see how it all fit together. And so some of the nucleotides from this model building are in museums that I've visited in London. And so it's kind of fun to see that. But they did a lot of their thinking and figuring by looking at structures and models. But we can't talk about the discovery of the structure of DNA without also including another important team member in all of this, who was Rosalind Franklin. And unfortunately, she died at the young age of 38. She spent a lot of her research time around X-rays, and I think she died of uterine cancer. So there's always been this talk about, did she die from cancer because of her exposure to X-rays, or what was the cause of her early death? But anyways, you cannot receive the Nobel Prize posthumously after you've died, so she was not included as part of the team. But she was a very skilled X-ray crystallographer, as they call it. And I don't know, honestly, a lot about how all of this works, but basically you can take the structure of a molecule, and if you bombard it with X-rays, those X-rays are deflected by the shape of the molecule. And so sort of the deflection pattern of those X-rays that's left gives you some hint about what that molecule looks like. And I mean, that's the extent and the depth of my knowledge about how X-ray diffraction works. But anyways, Rosalind Franklin was a really good X-ray diffraction person. And you see her famous picture down there in letter C. And to a skilled person in X-ray diffraction, that pattern suggests a double helix or some sort of helical structure. And so her work, coupled with the model building that Watson and Crick and Wilkins did, really came together then to allow them to come up with what we now know and understand as the structure of DNA. And I had to include this photo. This was, I don't know, I'm going to totally nerd out on you all, but this past January, maybe some of you had heard, Dr. Bryant in the biology department and I took a group of students to London, Scotland, some parts of the UK to learn a little bit about the medical history. And so after Watson and Crick really thought they'd figured out what the structure of DNA was, they left the laboratory where they were working, which was right around the corner from the Eagle Pub. And as lore has it, they sat in the booth where you see me and Dr. Bryant sitting and announced to everyone in the pub that they think they'd discovered, the secret to life, or they discovered the structure of DNA. And so at this booth, there's these two plaques. One of them just commemorates, that whole discovery of DNA, and another one commemorates the contribution of Rosalind Franklin. And Dr. Bryan and I and our group of students were fortunate enough to go to this pub and sit in that little bench. Oh my gosh, I was so excited when we were in Cambridge, England this past January. So just an exciting time for me in my life, but it's a little bit of the fun history of how DNA structure was determined, and I got to sit in the very seat where Watson and Crick sat when they announced it. So I don't know, it's just a little bit of fun. Okay, so what do we know now about the structure of DNA? I think we saw this slide before a long time ago, maybe at the beginning of the semester. And so this is just to remind you that if we start at the bottom of this, you see those little shapes, you all should be familiar with those. This is what we understand as chromosomes, 2 chromatids per chromosome there at the bottom. And the DNA then is tightly wrapped around proteins, and these proteins are called histones, and DNA wrapped around these proteins is what we call chromatin, chromatin. And we know that DNA is 2 strands held together by hydrogen bonds, and it takes on this double helical structure. And each strand is composed then of a number of nucleotides that are joined together in sequence. And each of those nucleotides then, you've got A, G, C, and T bases attached to them. So let's just quickly remind ourselves of the nucleotide structure. We've seen the next couple slides before, but I want you to make sure that you go back, review, and rethink about nucleotide structure, because it's going to matter as we start talking about how DNA is replicated. So as I said, you've seen this slide before. So here's a little closer look to remind you of the DNA nucleotide. And we've got the deoxyribose sugar, and each of those red numbers there, 1' through 5', are indicating the different carbons of the sugar, if you remember. And the base, here you're seeing a T attached. The base is covalently linked to the 1' carbon. We count the carbons in clockwise order, starting at that oxygen with the one to the right of the oxygen being number 1. And so the 1' carbon is where the base is attached. The 2' carbon is where you look to determine whether it's a ribose or a deoxyribose sugar. And this is clearly deoxyribose because we see hydrogen. There's no hydroxyl group there. All sugars have a 3' hydroxyl, which is really important as we start talking about DNA synthesis. You'll see that in a minute. And then you have your 4' and your 5' carbon is where your phosphates are attached. So I encourage you again to go back and review the nucleotide structure, because this is going to be important that we remember all of those carbons and what's attached where as we start to talk about DNA synthesis. Okay, so as we put nucleotides together then to make, in this slide, a single strand of DNA, we remember that the nucleotides are joined together by phosphodiester bonds, or what's shown here is a phosphodiester linkage. And what you notice, if you look at this single strand, is at the top we see the 5' phosphate of that nucleotide, and at the bottom, at the other end of this strand, we see the 3' hydroxyl. And this is important because we think about, and we talk about DNA having a direction, and we talk about a 5' to a 3' direction. And so the 5' we're referring to is the nucleotide at the end with the phosphate attached to the 5' carbon. And the 3' we're talking about is the nucleotide at the other end of the strand that has the free 3' hydroxyl group. So that 5' to 3' idea is really important to keep in mind, and that's what we are talking about. We're really talking about what's attached at the 5' and the 3' carbons, which we saw in the previous slide. So now if we put a double-stranded piece of DNA together, we see 2 strands on the far right here, held together by hydrogen bonding between the bases. And I think we're all pretty familiar with the fact that A pairs with T and G pairs with C, and we'll talk about how we know that and what some of the science is behind that as well. But what I want you to notice here in this slide, and you can see it in the corkscrew shaped structure or in the other one on the right that shows more detail, but one of the strands, one of the strands at the top, if I look at the one with more detail on the right, one of the strands has the 5' end, so you see the phosphate is at the top. And if I go down that same strand, I see that's the 3' hydroxyl, or the 3' end. So that strand has 5' at the top and 3' at the bottom. And if I go to the opposite strand, what I see is that strand is in the other direction, right? I see the 3' hydroxyl is at the top, and the 5' phosphate is at the end. And so what we say when we talk about this is that the 2 strands in a double-stranded DNA molecule are anti-parallel. In other words, they run like almost 2, lanes of a highway, 2 opposite running lanes. So one of the strands will always have the 5' to 3' direction if you looked at it from top to bottom, and the other one will always be the reverse. It'll be 3' to 5' if you look at that from top to bottom. And you can see that in both of the diagrams shown here. So the 2 strands are anti-parallel, and then we know that A always pairs with T and G always pairs with C. And so again, remembering that 5' and 3' and the anti-parallel nature of DNA is going to become really important when we continue here talking about DNA replication. And so in the next slide, I want to say a few things about some of the experiments that were done that helped Watson and Crick determine the structure, because these experiments showed that A pairs with T and G pairs with C. So let's talk about that for a minute. So there was another researcher named Chargaff, and he had done a number of experiments where he was a biochemist, and he was analyzing the amount or the proportion of these bases in the DNA from various species. I mean, you see in this table here the base content from a whole bunch of different organisms. So we've got everything from bacteria to humans, we've got fish, we have turtles, single-celled, other single-celled organisms. And so these are some of the data that he collected. And what you notice if you look at this table is the proportion of A is very much in line with the proportion of T, and the proportion of G is always pretty much equal to the proportion of C. And so that's kind of difficult to see in the top one with E. coli because they all look to be in similar amounts. But if we go down to just look at humans, you see a 30% proportion of A and 30% about of T, and then you see about 19 to 20% of G's and C's. And so these are what we call Chargaff's rules. And so Watson and Crick and Wilkins, when they were doing their model building and looking at the X-ray diffraction results from Rosalind Franklin, they knew this. Chargaff had shared these data with them, and so they incorporated this into their model building, thinking about, well, what kind of arrangement of DNA would support these kinds of proportions here. And of course we know now it's because if there's an A on one strand, then there must be a corresponding T on the other, and if there's a G on one strand, there must be a corresponding C on the other. So these again are collectively known as Chargaff's rules. Okay, so I'm going to kind of cut to the chase here and tell you sort of the results of the experiment first, and then we're going to go through an experiment. So this slide nicely shows DNA replication, and we know that DNA replication, we're going to talk about kind of how the overall mechanism of how DNA replication works, And we know DNA replication works through a mechanism that we now know as the semi-conservative mode of replication. And so if you look at this slide here, in both of these panels, you start out with a double helix, and both of the strands are shown in red. And again, you can see that anti-parallel nature that I talked about before. And you can also see that G and C are paired and A and T are paired between the two strands of DNA. And so what happens, you can see in blue at the bottom, as DNA gets replicated or synthesized, what's done is a method that's called semi-conservative replication. So the old strand, which is indicated here in red, is used as sort of a template or a guide to synthesize one new strand, which is shown here in red. So, or I'm sorry, shown in blue. So when you go from a single strand to two, or a single molecule to two molecules during S phase or DNA replication, the new DNA is half old and half new. In other words, it's half red and half blue. So that's what we call the semi-conservative method. And so what I want to do is take a few minutes to talk about, how was this ever discovered? Because I think that this is an interesting and a really great illustration of how the scientific method holds. So let's talk a little bit about the experiments that were done that supported this semi-conservative mode of DNA replication. So these experiments were carried out by two researchers, Messelson and Stahl, and I believe this was long about like 1958. So these experiments were done quite a long time ago. And again, what's the take home from this? Well, they were the ones who really showed beyond a doubt that DNA was replicated through a semi-conservative fashion in the way that was shown on the previous slide. So let's walk through first their experimental setup and what did they do, and then we're going to look at the results and some hypotheses and see their results, and then see which hypotheses were supported by the results that they gathered. So they were working with really simple organism. They were working in bacteria. And the first thing they did, shown here in the panel number 1, is they grew bacteria. Bacteria goes through, DNA replication just like any other organism. And they grew bacteria in medium that contained a heavy isotope of nitrogen. Okay, so we talked about isotopes before. Remember, they have different numbers of neutrons. And so this is nitrogen, but it's nitrogen 15. So it's a heavier isotope. And remember, there's a lot of nitrogen in DNA. All those nitrogenous bases, the A, G, C, and T, have lots of nitrogen in them. So if you grow bacteria in a medium that has nitrogen 15 in it, that nitrogen 15 is going to get incorporated into the DNA. So we're going to call that nitrogen 15, we're going to call that heavy, or heavy medium. And so they grew bacteria for many, many generations in this medium and got all of the DNA in that bacteria to have the heavier isotope of nitrogen incorporated into the DNA. And then at various times, after they'd grown the bacteria for a number of generations in nitrogen 15, at various times they would transfer it to media that had the lighter isotope of nitrogen, so nitrogen 14. And they let that grow for times that were in line with 1, 2, or 3 generation times. So one round of DNA replication, they put it in nitrogen 14 for that long, or 2 rounds of DNA replication for those bacterial cells. So they grow it in nitrogen 15, get all the DNA in the heavy form, and then they move some of those cells into nitrogen 14, where any new DNA now being synthesized is not going to have nitrogen 15 in it anymore. It's going to have nitrogen 14 in it. And then they isolated the DNA from those bacterial cells, and they could spin them in a centrifuge in this special gradients, cesium chloride gradients. And what you're basically doing when you spin is you're separating the DNA by its density. So remember, the nitrogen 15 we're going to call the heavy, and the nitrogen 14 we're going to call the light, and the light DNA. And so you spin it at high speeds for a long period of time in these centrifuges, and what happens is the heavier DNA actually separates out toward the bottom of the tube, and the lighter DNA, that containing the nitrogen 14, would separate out more towards the top of the tube. So you can get an idea of, is the DNA in these cells all containing the heavy isotope of nitrogen? Does it all contain the light isotope of nitrogen? Does it contain a mixture? So this is the experimental design, or the experimental setup, that Messelson and Stahl used. So now there are really 3 hypotheses that they were testing. And this first one, like I said, I gave it all away at the beginning, but this is the hypothesis, the semi-conservative mechanism of DNA replication. This is the hypothesis that their data supported. But I want to go through this and show you how and why their data supported this hypothesis. So for this, let's just look at this diagram in some detail. So I've put a legend over on the left-hand side. So all the heavy DNA that was made while the cells were in the nitrogen 15, those are the red strands that are shown in this diagram. So we start out at the beginning with all of the DNA being that heavy nitrogen 15 containing DNA. And then remember, for certain times, they switched it to the nitrogen 14 containing medium. And any DNA that was made while it was in the nitrogen 14 now is going to incorporate that lighter isotope of nitrogen, and that DNA is what's shown here in blue. Okay, so the heavy strands are red, the light strands are blue. And if the mechanism of DNA replication is what we call semi-conservative, what we would predict is, after the first round of DNA replication, one of the strands would be heavy, or of that kind of the old DNA strand, and then the newly synthesized strand would be light, and that would be what's shown here in blue. So we would expect after one round that the DNA that was made would be half heavy and half light, if the semi-conservative hypothesis is supported by their data. Okay, and we're going to come back, and I'm going to have you come back and look at that second round of replication that's shown here. But if semi-conservative replication is what's really happening, after one generation or one round of DNA replication, the new DNA should be half heavy and half light. But again, Messelson and Stahl did not know at the time that the semi-conservative method was, the one that their data was going to support. So there are a couple of other hypotheses. So this is another hypothesis that needed to be tested, and that is that the mechanism for replication was what we call conservative. And in conservative replication, instead of having, after one round of DNA replication, that you have a mixture of half heavy and half light, this hypothesis, if supported, would predict that after one round of DNA replication, you have one molecule of DNA that is all heavy. Right, so that is the same as the original double helix, and then the new one is all light, and so both strands are synthesized new, but you hold on and maintain that old one. So what this would predict then is that one of the newly synthesized molecules of DNA after one round would be heavy, shown here in red. Both strands would be heavy, and then the other would be blue. Both strands would be light. Okay, so this is the conservative mechanism. This would be the hypothesis that we are putting forth, that it would be that DNA would be replicated through a conservative mechanism. Okay, and again, we're going to come back and talk later about that second round of replication. But there's one more hypothesis that they could test with the experimental setup that they did, and that is to test the dispersive mechanism. Okay, and the dispersive mechanism is illustrated here. Well, what if what happens during replication is that you don't get a whole strand synthesized new? What happens if what you have are a mixture of some old and new, some blue and red, some heavy and light, sort of interspersed in the newly synthesized DNA. So at the first round of replication there, what you'd see is this mixture on both of the strands of the double helix. We see some red and some blue. And again, what would you expect to see if you were analyzing this? Well, it looks like, again, you'd have this mixture of kind of half heavy and half light DNA after the first round of replication. It's not all heavy and it's not all light, but it's kind of a mixture. So let's jump ahead and look at what results did Meselson and Stahl actually get by doing this experiment, and then go back and talk about which hypothesis, if any, were supported or rejected by the data that they acquired. Okay, so this is the results of their experiment. Okay, so let's look at a summary here in this slide. And so what I want you to do is pause and take a second. So here are our three different hypotheses: semi-conservative, conservative, and dispersive. And after the first round of DNA replication, we just kind of looked at this. But I want you to go back to those other slides where the DNA was laid out for each one of these hypotheses. And I want you to think about what would they have seen in the tube if semi-conservative was supported, if conservative was supported, and if the dispersive hypothesis was supported. And the other tube that I have here in that column labeled after first round of replication, that's just your control. Okay, so you have to have a control so you know where the light DNA is and where the heavy DNA is so that you have something to compare it to. So take a second and draw a line across those empty tubes there with the question mark after first round of replication. Where would you expect to see the DNA? If semi-conservative was supported, if conservative was supported, and if dispersive was supported. So take a second, think about it, and then we'll talk about it in the next slide. Okay, so in this slide, we've drawn the expectations for each of the hypotheses in those tubes that were empty on the previous slide. So if we go through each of these, so remember for the semi-conservative replication, after one round of DNA synthesis, we would have expected DNA molecules that were half heavy and half light. So those molecules would not be all light or all heavy. They would all be of sort of an intermediate density. And so we would draw a line somewhere between the light and the heavy. And the same is true if we jump down to the bottom for the dispersive method. Because for the dispersive method, remember that is where we had heavy and light kind of interspersed throughout each of the strands. And so we would see that same thing. No molecule of DNA after one round of DNA replication was light or heavy. It was a mixture of light and heavy. So we would expect, again, that sort of intermediate density that we see with a line drawn between the light and the heavy in the controls. But for the conservative method, if we remember how that worked from the previous slide, that's where the newly formed molecule is all light. So we had a whole molecule, both strands were red, and another whole molecule where both strands were blue. So if the conservative method was supported, we would see two bands because we'd have some of the molecules being light and some of the molecules being heavy. The newly synthesized ones would be all light. And so we would expect a result like that shown here. And when we did look at Meselson and Stahl's actual results in that panel that was labeled 1.0, what we saw was something in between the heavy and the light. Therefore, we can reject the conservative hypothesis because the data that they acquired did not support that conservative hypothesis. So what I want you to do next is to, again, go back to those three slides that showed the different hypotheses and diagrammed the DNA and what we would expect. And now pay attention to what the DNA would look like for each hypothesis after 2 rounds of DNA replication. And now fill in those tubes there, the empty ones, under the second round of replication column with the question marks next to them. Where would you expect to see the lines after 2 rounds of DNA replication for each of the hypotheses? And then go back to the results that Meselson and Stahl actually got and look at the panel labeled 2.0 and see which one of the hypotheses was supported by their data. And I think what you'll see is you can then reject the dispersive model. And their data supported, of these three models or three mechanisms, their data supported the semi-conservative model of DNA replication. Okay, so we know DNA is replicated in a semi-conservative method. We know the structure of DNA. So now let's talk a little bit more about the process of DNA replication. So I'm going to divide this into two sections, the process, and then I want to talk about the actual enzymes and other things that are involved in the process. Okay, so let's first talk about the process. DNA replication begins at points along the DNA that are called the origins of replication. And you see that shaded in blue here at the top of this diagram. And so the DNA replication begins at these origins, and it starts out in a bidirectional fashion. In other words, the DNA gets synthesized. You can see in the middle panel here where we're now starting to draw in the blue lines. So DNA synthesis actually occurs in both directions out from what we might call this opening or this bubble in the DNA. And so that establishes what we call replication forks. And so we're going to talk about that in a bit more detail too. But DNA replication is bidirectional. It goes out in two different directions from the original or the origins of replication. And so this is what it might look like in this slide, an origin of replication on a linear chromosome. And in the next slide, remember, bacteria have circular chromosomes, but the same is true, an origin of replication, and it also goes in a bidirectional fashion. So this is a diagram of how DNA replication would start and go on a circular chromosome in a bacterium, for example. So again, you have a blue shaded area indicating the origin of replication. DNA replication goes out bidirectionally, so it would go out in opposite directions and eventually kind of come around and meet in the middle. So this is how it would work in a circular chromosome like we would find in bacteria. One other thing to point out about origins of replication is that on linear chromosomes, long linear chromosomes like those you would find in most eukaryotes, there are multiple origins of replication. So replication doesn't start on one end, for example, and go all the way through to the other end. It starts at multiple places throughout the entire length of the chromosome. In bacteria, where you have a circular chromosome, it generally starts, there's only one origin, and it makes its way in both directions around the circle. But on linear chromosomes in eukaryotes, you have multiple origins of replication, as is shown here with the multiple blue shadings on the top of this slide. So here is one of those origins of replication where the two strands of DNA open up, or again, sometimes they're referred to as kind of these bubbles that form. The strands separate, and you can see in the boxes here, there are two places that we call a replication fork, right? Where you have, I don't know why it's called a fork, but there are two of them. And the one on the right is going to move, and DNA is going to be synthesized going to the right side of the slide, and the one on the left is going to have DNA being synthesized on the direction to the left of the slide. And so we have these replication forks that start the actual synthesis or the elongation of DNA replication. Okay, in this slide, what we're looking at is half of the bubble that we saw on the previous slide. So we're taking that replication bubble and we're cutting it in half, and we're only looking at one of the replication forks, the one on the left in this case. And so DNA replication is going to proceed to the left of this slide. So there are a couple of things that I want you to note from this slide. So remember, there's a few things we've already established. We know that the two strands of DNA are anti-parallel. If you look at the red strands shown here, there's a 5' end and a 3' end to both of these red strands, but they run in opposite directions. So we know that. We know that DNA replication is semi-conservative. Messelson and Stahl showed us that, which means that one of the old strands is used as a guide to help us determine what nucleotides, the order of the nucleotides, go in for the newly synthesized strand that's shown here in blue. And then the third important point that we need to remember that really dictates how DNA replication works is that new DNA, the blue strands, are always synthesized in the 5' to 3' direction. And you can see that here with the blue arrows. They're always going 5' to 3'. And so because of those three things, semi-conservative replication, DNA's anti-parallel, and synthesis of new DNA always has to go in the 5' to 3' direction, the way the two strands are synthesized is by definition different. And so one of them you can see at the top blue one is called the leading strand, and the one at the bottom is called the lagging strand. And so we use those two different terms to talk about the two different ways that these strands have to be synthesized. And so we'll look at that in some more detail then in the next slide. Okay, so as DNA synthesis proceeds, so the first thing I want you to note, so remember the leading strand is on the top, that's the long blue one, and then the lagging strand is shown now in two fragments on the bottom. So first of all, what are those little yellow bits? Well, the first part we have to remember is that in order for DNA synthesis to occur, there needs to be a primer that's laid down first. And so those little yellow segments are primers, and we'll talk some more about that in a little bit. But the leading strand is always primed. You can see the little yellow band or little yellow part is back at the origin of replication. So the leading strand is primed at the origin, and it is synthesized in a continuous manner. So once it starts synthesizing DNA, it can just continue to move, and as the replication fork moves and the DNA unwinds, as we move towards the left in this case, that synthesis just continues as more of the single strand from the red molecule here gets exposed, the blue strand just gets laid down, and we see a growing strand for the leading strand that moves toward that replication fork. But on the bottom, things have to work in a slightly different way. So what happens with the lagging strand is that it's primed at multiple places. Because synthesis has to happen in the 5' to 3' direction, and this replication fork, or the DNA, is unwinding in the opposite direction, we have to synthesize the DNA in these little fragments that are called Okazaki fragments. So we're still going 5' to 3'. Each one of the segments has to be primed with the little yellow primer, but we synthesize the DNA in small segments going 5' to 3', and so the growing strand actually moves away from the replication fork. So the arrows for the lagging strand are pointing away from the replication fork. And as DNA replication then continues, what you see in this slide is the leading strand continues to extend as that DNA continues to unwind at the replication fork. So the leading strand again continues. It's a continuous synthesis in the 5' to 3' direction. But what we see with the lagging strand is that the Okazaki fragments now get physically joined to one another. So the primers will eventually get removed, and the Okazaki fragments literally get kind of glued together to give us one continuous strand when we're talking about the lagging strand. So that is kind of an overview of the process of how DNA replication works. You have a leading strand and a lagging strand, and all of this is just because of the way the structure of DNA is laid out. So what I want to do next is we'll go through sort of this process again, but I want to highlight some of the proteins and the enzymes that are involved in making this all happen. So what we're looking at here again is a replication fork, same kind of thing that we were looking at previously, but now I want us to highlight and look at what some of the proteins are at the replication fork that are allowing a lot of this to happen. So first of all, we see the DNA helicase. So there has to be a protein involved in unwinding the single strands. And so helicase goes through and it literally breaks those hydrogen bonds between the A's and T's and the G's and C's. And it's really helicase that unzips, if you will, the DNA strands as we move through and open up that replication fork. So DNA helicase separates those two DNA strands. And then another protein shown in this slide is called the DNA topoisomerase. And remember, DNA is a helix. It's a twisted coil. And as we unwind, what happens is you get a lot of torsional strain, and it just kind of gets all twisted up. And so topoisomerase works to untwist those strands so that the DNA doesn't break. As it unwinds, topoisomerase is important for making sure that it gets untwisted as it unwinds. And then the third important protein that we see here are those brown ones that are attached to the single strands, called single strand binding proteins. And they are responsible for just holding the bubble open, right? We want DNA replication to occur without the two strands of DNA coming back together and reforming a double helix. So these are three important players at the replication fork that are unwinding the DNA, untwisting the DNA, and holding the two strands of DNA apart from one another. And of course, probably the most important enzyme and player in all of this is the DNA polymerase, or the enzyme that actually does the synthesizing, actually catalyzes the joining of one nucleotide to another. And so there are two things that are important about the activity of DNA polymerase that are highlighted in this slide. And one of them is that in order for polymerase to begin synthesis, it needs a primer. In other words, it can't just jump onto a single stranded piece of DNA and start synthesizing the other strand. It needs a little start. And so DNA polymerase does require a primer. And as we said earlier, because of some energetic constraints, DNA synthesis always has to occur in the 5' to 3' direction. And you can see that in part B on this slide. So the new nucleotides, which are indicated there in blue, are always added to the growing 3' end. And that is one of the requirements of DNA polymerase. And we'll look at that in a little bit more detail here in the next slide. So at the top, again, we're looking at DNA polymerase and how it works to bring in new nucleotides. And at the bottom, we are looking at, so again, the chemistry of DNA replication. So you see a nucleotide, a C, being brought in. And when nucleotides are brought in, they actually are triphosphates. And so they come in and they physically then form that polymerase is important for catalyzing the formation of that phosphodiester bond. And in the process, 2 phosphates then are lost because we have 1 phosphate then as part of that phosphodiester bond. And so this is why it grows. You always attach a new nucleotide to that 3' OH of the nucleotide that's already existing in the blue strand. So we always have 5' to 3' DNA synthesis. New DNA is always made in the 5' to 3' direction. So here are a few more details. This is just a larger diagram of the smaller one we saw in the previous slide. So here's the polymerase. Again, it needed a primer to get started, but what it's doing is it's reading the template strand. And the template strand tells it whether it should put an A, a G, a T, or a C in next. But you can see the incoming nucleotides with 3 phosphates are always being added in that 5' to 3' direction. So the blue strand is being added to the 3' direction, but of course, remember DNA is anti-parallel. So the template strand, the old strand, is being read from 3' to 5', but the new DNA is being synthesized from 5' to 3'. And if you remember, I said that polymerase always needs a primer. So another important protein and enzyme in this whole process is an enzyme called DNA primase. And so this enzyme lays down about 10 nucleotides, and these are actually RNA nucleotides, to sort of get things started for the polymerase. So you can see the leading strand at the top, we lay down the primer back at the origin, and the lagging strand at the bottom, we're starting to prime for the synthesis of the first Okazaki fragment. So RNA primase lays down small little pieces of RNA that then the DNA polymerase can latch onto and continue synthesis. And these little primers are later removed and replaced with DNA as DNA replication proceeds. So here is a nice summary of how all of this is starting to kind of come together. So we see the leading strand at the top, the lagging strand at the bottom. We have the yellow primers being put in place, and the polymerase now polymerizing the new blue strands, leading strand in a continuous fashion, moving in the same direction as a replication fork, and then the lagging strand being synthesized in small Okazaki fragments that will later be joined together to form one long lagging strand. And this just continues to show the process as we move down, as that replication fork opens and as the DNA becomes unzipped. The leading strand, the polymerase, just keeps moving down that leading strand and incorporating new nucleotides in that 5' to 3' direction. And the lagging strand on the bottom, the polymerase continues to synthesize new Okazaki fragments, and the primase continues to lay down new primers. And the polymerase then removes those RNA nucleotides. The yellow primers get removed, but then those two, the Okazaki fragments do need to be joined together. And so there's one more enzyme that actually does that and joins those Okazaki fragments together, and that's shown in the next slide. So in the end, we need another enzyme called DNA ligase. And what DNA ligase does, shown here in green, it actually catalyzes the formation of the phosphodiester bond between Okazaki fragments. So that when all is said and done, we'll have all of these small Okazaki fragments joined together or glued together by the DNA ligase. And the leading strand, of course, just synthesizes continuously, and that polymerase just moves along as the replication fork opens.